In this video we're going to cover basic cell structures and a little bit about transport. So let's zoom in on this cell a little bit so we can get a better look at it here. And so this is sort of a generic bacterial cell, so it's a prokaryote. And so we're going to start in the center, right? So we're going to start in here. And this is what we call the nucleoid. Alright, so there's no membrane-bound nucleus, but there is a chromosome here. There's only one copy of the chromosome, so it's haploid, alright, unlike our cells, which are diploid. This structure here, that is also in purple, is DNA also, but it's called a plasmid. It's extra chromosomal DNA. We'll talk about more about those later, but they tend to have specialized genes on them. The little red dots everywhere are ribosomes, and those are of course the center of protein synthesis. And then we've got inclusion bodies where the cell is storing compounds for later. Alright, and so that's the basic sort of internal structures that we're going to find. Then we're going to have a wall, alright, and so we've got a plasma membrane and a cell wall right along here. And then this bluish green teal color, I guess you could call it. This is called a capsule, and that's a polysaccharide layer, and it is very slimy, very gunky, alright, and it helps protect the bacteria from drying out, changes in pH. If it's a pathogen, it helps prevent your immune system from engulfing it. And then these little hair-like structures that we're seeing spread all over, these are called fimbriae. They're made from proteins, and they help the cell attach to specific surfaces. So think of an intestinal bacteria, right? It would need to grab onto the intestinal lining, otherwise it's just going to get flushed through the system, right? And so those are protein structures. And then over here we have a plasmid, which is sort of like a giant fimbriae, so it's also a protein structure, and its role is actually to exchange DNA with other cells, and then we'll talk about it later. I think that's all the structures. So one of the nice things about prokaryotic cell is it's very simple. Now bacteria come in different shapes, alright, and so you will want to remember or memorize some of these shapes, alright, and so round is referred to as coccus, alright, and so cells like staphylococcus, streptococcus, they are round in color, alright, or round in shape, rather. Bacillus are rod shapes, alright. Vibrio, such as Vibrio cholerae, they are a curved rod, and you can see the curve on this cell right here. All right. I don't really care about the Coccobacillus or even the Spirillum so much, but I am interested that you know the spirochete, which are very corkscrew in shape, and these are important for certain diseases, and so we'll talk about them later. The most famous, of course, being syphilis is a spirochete. Alright, so just to review real quick, you're responsible for coccus, bacillus, vibrio, spirochete. Now the bacteria also, in addition to having specific shapes, they can grow in sort of cluster patterns. All right, and so there are a few that are important here, mainly from a disease perspective. So when we see things that are diplococci, such as this one here, all right, Streptococcus pneumoniae is something that does this. All right, Streptococcus pyogenes, that causes strep throat, grows in long chains of cocci. And then staph tends to grow in more of a grape-like cluster. So if we're finding the infection has more of this pattern of growth, we would suspect staph over strep, for example. All right. Okay, so the membrane. So we know a number of things about the membrane, right? You've learned these in general biology classes, AMP, and so on, right? That it's this barrier to the environment. And so in doing so, it separates the cell, right, the internal contents of the cell, from the environment that surrounds it. And so we often talk about the types of solutions that we find our cell in. So let's zoom in. Here we are. Right? And so what we're going to start with is what we call an isotonic solution. All right, and so this is where the solution, right, on the outside has the same concentration of solutes as the inside of our cell, and so water then will move across the membrane, right, in and out in equilibrium, and so there will be no change really in the net movement of water across our membrane here. So animal cells tend to live in isotonic solutions, so our cells are isotonic, all right, with their environment. Now, if we look at a hypertonic solution, okay, let's get that centered, all right, this is where we have a high concentration of solutes on the outside, all right, and a relatively lower concentration on the inside, and so now the water tends to move out, all right, and so the water moves out and our cell shrinks, of course, until the solute concentration on the inside of the cell becomes the same as the concentration on the out, all right. And finally, the world that bacteria live in is they live in what are called hypotonic solutions. So they tend to be, more concentrated in solutes on the inside and less concentrated in their surroundings. And they have a cell wall so that when the water presses in, right, so when the water comes rushing inside the cell, if that water gets, very concentrated in there and it pushes out, right, the pressure inside our cell starts to grow. It just pushes up against the cell wall and it's not an issue, right? So this is the world that bacteria live in, so they live in hypotonic solutions. Now, let's just go ahead and go back to this previous example here and talk about why this occurs. So let's look at the hypertonic solution. And the basic premise here is that as you add solutes to water, so this on the outside is very concentrated, you decrease the potential energy of the water. So we have low energy water out here, and then we have more dilute water on the inside of our cell, all right? And so lower solutes means higher water potential or higher potential energy, so the water moves from high energy to low, all right? So in this example, then, we have low energy water on the outside because there's more solutes, high energy water on the inside, the water flows out. In our example here, all right, we have very little solutes on the outside, so we have high energy water out here, right? We've got higher solute concentration on the inside, so we have higher solutes, and therefore we have lower energy, and the water moves from high to low in. Okay. All right, so in plant cells and in bacterial cells where we have a wall, all right, so here we have the bacterial cell wall. All right, so here's our isotonic solution, right? So the water's moving in and out at a constant rate, and what will happen is that that's when a plant wilts, right? It starts to wilt, it becomes flaccid. If you place the plant cell or even a bacterial cell into a hypertonic solution and it doesn't have a mechanism to protect itself, the water will exit and then the cell membrane will pull away from the wall and it becomes, basically it'll kill the cell. And here's where they like to be is in this hypertonic or hypotonic solution, So you have low solute concentration on the outside, high solute concentration on the in. The water moves from out to in. All right, so here's our nucleoid, excuse me, and so what we've got basically is a concentration of DNA in the middle of our cell. The chromosome in bacteria is circular, right, so it's just circular double-stranded DNA, like so, and it's all twisted up, which is why it's just kind of a big mess of spaghetti in here is really what it would kind of appear to be. It's just a supercoiled, twisted up rubber band, basically. All right? And so we don't separate then the activities associated with the DNA from the cytoplasm, right? It's all continuous. Now here's just a cartoon of the ribosome. All right, so if we assemble the ribosome in two subunits, all right, so we have a small subunit, a large subunit, and they come together to form the complete ribosome. All right, and they do so, and we would have basically our mRNA running through the middle here, and we would be building a protein that would be coming out like so. All right, and so these are our centers of protein synthesis. All right, another structure that's unique to bacteria, all right, is the endospore. And so I'm going to zoom in real quick just on this top part so we can get a better explanation of how this works. Okay, so it starts out and it appears to be cell division. So it looks like we've duplicated our nucleoid. But then what happens is that one of these nucleoids gets its own membrane, all right, and so now it's surrounded by two. And so you can see it here, and we call this the forespore. You don't need to memorize all these different stages or structures. But what happens then is once the spore has internally formed, right, so that's what we call it, the endospore, it's basically released from the cell, all right, and the original cell dies. So it starts out as cell division, but then it's asymmetric in that you only get one spore from the cell division. So it isn't true cell division, it's actually an aberrant form of cell division. Now if we look at the electron micrograph here, you can see it a little better, right? So here's my spore, right here, all right, and this structure is extremely resistant to environmental stress. Some spores can be boiled for a week and still germinate. Some of them have been trapped in amber for literally millions of years, and they'll still germinate. And here's a slide of some stained spores, and the spores are here in the greenish color on the inside of these cells. All right, so here's our membrane. Let's zoom in just a hair. All right, so the things that you should be remembering about a cell membrane are phospholipid bilayer, right? That we've got the hydrophobic tails on the inside, right? Polar heads on the outside. All right, we've got proteins, all right, that we call transmembrane proteins that reach all the way across, all right, the membrane. All right, then we have peripheral proteins which are associated with the surface. Okay, a glycoprotein simply refers to a protein that contains polysaccharides attached to it. All right, and on the inside of our cell here we've got a little bit of cytoskeleton, and bacteria do have a crude cytoskeleton. So we're outside the cell here, we're inside the cell here, all right, and that's our basic membrane structure. So how do we transport things across this membrane, right? It's a semi-permeable barrier. So one mechanism is simple diffusion. So we've got the extracellular fluid out here. We've got the internal or the cytoplasm here. We have a high concentration of whatever this solute is. We've got a low concentration on the inside of my cell. And so over time, as we work our way from left to right, some of these molecules will just simply work their way across this membrane. And yes, it's slow. They aren't very good at getting across, right? And only generally small things that tend to be nonpolar in fact. And so something like CO2, all right, it diffuses. Oxygen is something that diffuses quite readily. And so these are small molecules, and basically they will either have enough momentum, right? So if we look at this one here, if it can blast its way through, all right, it'll make it to the other side. All right, and so that's simple diffusion, high concentration to low, no energy added. Now, another mechanism is facilitated diffusion. Now, this is where we basically use a protein channel, right? So here's our carrier protein, all right, or our protein channel. And so what it does is it creates an environment that is conducive for the solute to work its way across our membrane. All right, and so in doing so, it goes down its concentration gradient again, so there's no energy required. So it goes from high concentration out here to low on the inside, and this channel basically facilitates the movement of this thing across this semi-permeable membrane, right? So it creates a channel or a hole that allows it to flow through, as opposed to it having to battle its way through the phospholipid bilayer. And finally, we have active transport, which is going to be from low concentration to high. And you should be familiar with this mechanism here. This is the sodium-potassium pump. All right. So we have two different concentration gradients. They're indicated over here on the far right. Sodium is going to go from low concentration on the inside to high on the out. Potassium is going to go from low on the outside to high on the inside. So here's how it works. All right, our sodium is the green molecule, our potassium is the red. All right, so when we're on the cytoplasmic side of the membrane, two sodiums bind to our transport protein. All right, there's particular binding sites for them. All right, then ATP binds to our protein. All right, and in doing so, we transfer a phosphate group, and you can see it right here. It's that tan, sort of house-shaped pentagon, right? So it's right here, and so this thing is attached to our transport protein, and that causes the sodium to be released to the outside of the cell. It changes the shape of our protein. The binding of the ATP does. Now, in this position where we're open to the outside, we can now have potassium bind, okay? And so when the potassium binds, all right, that will trigger the release of the inorganic phosphate, all right? And we go back to our original conformation over here that allows sodium binding, and when the sodium can bind, the potassium cannot, and so the potassium is released to the inside of the cell. Our energy source is ATP, all right? And we have moved two substances up their concentration gradient, right? Sodium from low to high, inside to out. Potassium, low to high, outside to in. And that is it. And those are the basics of transport, all right? Some structures of the cell. And so we'll cover some more of this in class.